Thanks for the introduction. Thanks for the invitation again. First talk I want to give is a talk about monoclonal antibodies and antibody-drug conjugates in breast cancer. And this is definitely a success story. And I think you know this already, but it's just to give you a summary of this success story, and this is to talk about. My agenda, give you some ideas how it worked out from bench to bedside. Then the combination of Trastuzumab and plus minus Pertuzumab in early breast and metastatic breast cancer, and then the conjugate TDM1 in metastatic breast cancer, second and further line. So this is what happened, and you always know that it takes a long time from designing a target or from detecting a target, and this was done with HER2 in 1981 to the first approval study, second, third, and then different indications. So this is something we talked about extrapolation, we talked about biosimilars. And in 1995, there was the first approval study in metastatic breast cancer by Dennis Slayman from UCLA. And then we got this approval for metastatic in the US, and then a second study for approval metastatic breast cancer by Marty, and then we had this adjuvant SHIRA trial, and then and so on and so on. So the story continued in the end. And in March 2013, we had the EU approval of Pertuzumab in HER2 positive metastatic breast cancer for first line. And then we had a subcutaneous formulation for Herceptin in August 2013, November 2013. So many things happened in that year. The EU approval of Kadcyla, TDM1, and then metastatic breast cancer for second line, and then Perjeta was approved in the EU for HER2 positive early breast cancer in the neoadjuvant setting, and now we're waiting for the approval of the combination of Pertuzumab and Trastuzumab in the adjuvant setting. Well, we know that HER2 status is associated with aggressive disease and poor patient outcome, and this was the goal we want to meet when studies were conducting to show significant benefit for the use of an HER2-directed treatment. And these are the patients. This is a very famous slide from Dawood and colleagues, published 2010. These are the patient population being HER2 negative, so the better subgroup. And this is the patient population being HER2 positive, so the more aggressive patient population after receiving Herceptin. And this is the patient population being HER2 positive without Herceptin. So you see there's a really highly significant difference if the patients in a HER2 positive setting were treated with Herceptin, Trastuzumab, or not. And there's really a number of lives saved after the introduction of Trastuzumab in first-line HER2 positive metastatic breast cancer from 1990 to 2013, and this is something many colleagues have fear of biosimilar Trastuzumab, that this still be reached, and I think with all the talks we gave before, with a careful dealing with the situation, we will do this. For early breast cancer, in a neoadjuvant setting, we have numerous trials showing that the dual blockade out of Pertuzumab and Trastuzumab led to a higher percentage, led to a higher pathologic complete remission. So this is over all the studies, and the PCR with a dual blockade is illustrated in the red bars in this slide. The first study showed a higher PCR with a twofold higher PCR in the HER2 positive hormone receptor negative patient population. In the neoadjuvant setting was the Neosphere trial. I don't show you this, the study design for the matter of time, but this is an update on the survival data, and there you see that PCR correlates with the progression-free and disease-free survival in this study with a 16-month follow-up. That means all the patients who received a PCR and received a higher PCR because of the dual blockade, and this is the blue line, the blue Kaplan-Meier curve, they received even a better survival benefit. The HANA trial was the trial testing the subcutaneous formulation of Trastuzumab in a neoadjuvant setting, and there you see the same. So PCR will lead to a survival benefit. What about the adjuvant HER2-directed treatment? We know that we have significant disease-free and overall survival benefit after 10 years of follow-up. We know that the recommended treatment duration is 1 year. This is better than 6 months. That was shown in the French FAIR trial. We know that the subcutaneous application with the same has the same efficacy compared to the IV formulation, and we have 2 studies showing this, the PREFER and the HANA study. We know that simultaneous application is possible with paclitaxel, docetaxel after ACEC, and in docetaxel carboplatin out of the BCRG-006 study. And then we know that simultaneous application is not recommended together with anthracyclines due to cardiac toxicity, and Trastuzumab plus paclitaxel, so mono Trastuzumab with just paclitaxel shown in the APG trial, was successfully done in T1, T2 stage, node negative, G1 up to 3, hormone receptor positive and negative. So this is something you can do with a lower tumor burden in the adjuvant manner. These are the studies. I don't want to go into detail because I just have 15 minutes. The HERA study comparing 1 year to 2 years, the BCRG-006 study with an anthracycline-free arm, and then the NCCTGN-9A31 and the NSABP-B31 study from the US. All these studies led to a significant difference in disease-free survival and even in overall survival, and these are the 10-year follow-up data. Moreover, Trastuzumab has a consistent safety profile, and we all had fear in the beginning, what happens to the HER2 receptor and the myocardium if I block the HER2 receptors there, and I definitely do this, and we had higher cardiac toxicity, but we know from the adjuvant studies that this cardiac toxicity is reversible when you stop the treatment, and therefore we have to monitor all our patients via ultrasound of the heart during the treatment. But this has consistency with approximately more than 1 million patients treated with Trastuzumab. The desperately awaited data were presented by Gunther von Minkwitz, a colleague of mine from the German Breast Group. He presented the oral presentation at ASCO 2017, so this year, and this study should answer the question, does it make sense to go for a dual blockade in the adjuvant setting? So from the NEO-ALTO study, it was successful, but now we wanted to show, is this done in the adjuvant setting by using the combination of Trastuzumab and Pertuzumab? So, of course, 2 arms, straightforward design, 4,800 patients. And this is the data, very conclusive. Yes, it was a positive trial. The Kaplan-Meier curves are really narrow to each other, but in the intent to treat primary endpoint analysis, and the endpoint was invasive disease-free survival, there was a difference of 1.7%. That means a significant difference. Not for the node-negative subgroup. So treating node-negative patients in this manner doesn't lead to a significant difference, but in a node-positive subgroup with a hazard rate of 0.77 and a delta of 3.2% regarding the invasive disease-free survival. Trastuzumab first line, okay, I don't have to tell you, I think, because this is data that is quite old, more than 50 years old. The Slayman study, the Marty study, led to an overall survival benefit. And then was the idea, can I add something more? Can I add in a metastatic setting something more for the use of Trastuzumab? Because we know that approximately 50% of the patients with HER2-positive metastatic progress within 12 months of first-line treatment. I showed you already the neoadjuvant and adjuvant data, but the story came from the metastatic situation. So the answer was, how can we improve upon Trastuzumab? We know that there are 4 receptors in the HER2 family, And these receptors, they can build heterodimers or homodimers. We know that HER2 is the most important HER2 receptor with different subdomains. And we know that HER2 is able to build heterodimers, and the most common, most potent oncogenic dimer in HER2-positive breast cancer is the dimer consisting of HER2 and HER3. And HER2, of course, and we know this, is a rational target for anti-cancer therapy. So pertuzumab was designed and detected, and pertuzumab is a first in-class HER2 heterodimerization inhibitor. It inhibits the dimerization between HER2 and the other HER2 and the other HER family members. And this is a cartoon just to illustrate. So this is trastuzumab binding on one side, this is pertuzumab binding on the other side, and then it inhibits the dimerization, the heterodimerization between the HER family and the HER2 receptor. And the first study showing a really significant benefit, and you may be aware of that studies in the metastatic setting don't lead in the end to overall survival benefit, but Cleopatra study led to a survival benefit because we identified the target, we identified the predictive factor, HER2 positivity, and then we could target this factor, and of course this may lead to, and this definitely led to a survival benefit. So what was done in this study? 808 patients, first-line metastatic breast cancer. Trastuzumab was compared to pertuzumab and trastuzumab, and chemo backbone was docetaxel for 6 cycles and more. And this was the result in a 15.7 months longer overall survival, and this was really a milestone in treating metastatic breast cancer. What do we do with the triple positive patients? We know that there is a crosstalk between the HER2 signaling pathway and the estrogen receptor signaling pathway. And if you shut down one pathway, the other pathway is still active. So what do we do when we want to, inhibit both pathways? And for patients who are hormone receptor positive and are not able to get chemotherapy, we have different options. So these are studies with trastuzumab and NAI. There's one study with lapatinib and NAI. But nevertheless, the synergism of chemotherapy and trastuzumab is even better than the synergism you get with an endocrine treatment and trastuzumab. Well, what is new since San Antonio Breast Cancer Symposium last year, so December last year, and we all are traveling to San Antonio in 2 weeks. We had data from the PERTAIN study, and the PERTAIN study examined the dual blockade of pertuzumab and trastuzumab plus an AI versus trastuzumab and an AI, and this led to a significant difference regarding progression-free survival. So there is data, and you can use dual blockade plus an endocrine treatment. So what about the trastuzumab conjugate TDM1 and second line? What happens? So trastuzumab binds to the HER2 receptor, and there is a really stable link of myotensin, so this is a cytotoxic molecules, and then the receptor is internalized in the cell, and there the linker disappears, and the cytotoxic compound can lead to a benefit and lead to apoptosis of the cell. We have different studies, but the approval study was the Emilia study showing an overall survival benefit in the confirmatory analysis. And capecitabine and lapatinib, the standard of care to that time, was compared with TDM1. And there's really a big difference with a hazard rate of 0.68. And for the patients having CNS metastasis, and quite a few of the HER2 positive patients get CNS metastasis, we had an additional overall survival benefit. I mean, patients with CNS metastasis. So this was, to me, a really important, significant difference. And this is something what led to TDM1 as a standard treatment for these patients. What do we do with TDM1 in further lines? So the Teresa study tested TDM1 after heavily pretreated patients in comparison to a treatment of physician's choice. You could use different kinds of chemotherapy, and you may read that the patients were really extensively pretreated. A third of the patients received more than 5 treatment lines before they received T-DM1, and despite this extensive pre-treatment, patients received a significant benefit for progression-free survival in this situation of nearly 3 months and a reduction of risk of progression nearly by 50% in comparison to treatment of physician's choice. And what else can you do if you have a patient heavily pretreated, not able to receiving a chemotherapy? Then you can combine Trastuzumab and Lepatinib. From the Blackwell data, we have an overall survival benefit and a prolongation of the overall survival of more than 4 months. What else was done? The MARIANNE study was done, and this was logically because the treatment success that was received with T-DM1, the treatment success that was received with Trastuzumab. So this study was designed to compare T-DM1 and T-DM1 plus Trastuzumab and the standard of care of taxane and Herceptin, Trastuzumab, but unfortunately nothing was different, and there was not a significant difference for the T-DM1 plus Trastuzumab arm, but the endpoint of the study was showing non-inferiority, so not superiority. So in the end, it was a positive study because the endpoint was reached. There was a non-inferiority of T-DM1 versus Trastuzumab and taxane, and T-DM1 had the same efficacy but less toxicity. So if you think of a treatment of Trastuzumab and taxane for the patient you don't want to have that toxicity, you can choose it. You can choose T-DM1. And the overall conclusion was T-DM1 is as good as Trastuzumab and taxane. T-DM1 plus Trastuzumab is as good as Trastuzumab and taxane. And then the data we received from the Cleopatra study, Trastuzumab and Trastuzumab plus taxane is in favor to T-DM1. Yeah, thanks. Again, my team from Frankfurt, thanks for your attention. And I will need to go over to the second talk. Now you have to help me because I'm a Mackie. So the second talk I was asked to give is, well, attacking target mutations. Our approach is promising, and as I mentioned before, I'm a gynecological oncologist, so therefore my field is breast cancer, ovarian cancer, endometrial and cervical cancer, but I'm a known specialist in Germany for breast cancer, so therefore I will focus on the target mutations we have in breast cancer treatment. Well, this is a German slide, but in the end it illustrates what happened the last year. So we had one treatment, and we had a heterogeneous illness, cancer. And we expected by giving the same treatment to all subpopulations that we will succeed. No, not in the end, because we have very good efficacy, and then we have no efficacy, and then we have toxicity. So we have to adjust it that we have an heterogeneous illness. So therefore we have to treat these patients with different treatments adjusted to predictive factors, biomarkers. Liquid biopsy will come in I hope within the next 4 or 5 years. But this is something we learned. And therefore, oncology 2012 and 2017 is not consisting of chemotherapy compounds. It's consisting of biologicals. And now, remember what we told about biosimilars. So the biosimilar world will be like a tsunami wave. You think of all the biologicals that are under intensive evaluation and are approved when the patent will expires. This is what we know from breast cancer. So we know there are luminal-like cancers, there are triple-negative basal cancers, and there are 2 positive cancers. And we know that from the phenotype, we can treat these different subgroups, of course, different. And we have to treat them different because they have a different rate of aggressiveness, and they have different targets we can treat. But starting with the problems. So when we identified the triple-negative breast cancer, and we know, yes, offer a BRCA test, neoadjuvant treatment is preferred, you use anthracyclines, taxanes, and so on. But then he got this idea of the triple negative is not triple negative, because from intertumoral heterogeneity and and triple negative breast cancer subtypes from gene expression, we have this idea that we have 7 different subtypes. What does this mean? If you do a clinical trial for triple negative breast cancer containing of 15% of all breast cancers, dividing by 7 subgroups, then you can imagine how high your sample size would be designing a proper trial in this situation. And this matters because in the middle, the red bar, the LAR subtype, so this is the androgen receptor positive subtype. And there you need not chemotherapy because this is a PCR. So these patients didn't receive a PCR by neoadjuvant therapy. This was the PRECOG study presented a couple of years ago in Chicago at the ASCO. So you have to get an idea to define your responders and non-responders, and at the moment studies are going on with androgen receptor inhibitors in a matter of a window of opportunity trial. So you give an androgen receptor inhibitor for a couple of weeks, and then you do a rebiopsy, and then you get this idea, is it worth to go for an androgen receptor inhibition or not? So this is not for clinical routine yet. So do targeted mutations help? And this is by Fabrice and Jay, and I really like this guy coming from France, and what did he do? What did he do? He had a patient set of around 400 patients, so exactly 423 patients, and he took a biopsy of every metastasis of these patients. And then he had to realize, okay, I just have 404 patients where I get a biopsy of the metastasis. And then he did a genomic profiling. This was possible in 287 patients. And then, so because we talk about target mutations, just 25% of the targetable mutations could receive a treatment that existed. So therefore, from the 423 patients, just 12% could be treated because of a target mutation with a medicinal product that existed. And then you are interested as a physician, so what is the clinical benefit rate? At least you need a clinical benefit rate of 16 weeks, more than 16 weeks, and then we had to realize that just 3% of the patients could receive treatment. They had a target mutation that led to a clinical benefit rate of more than 16 weeks. So, I feel very sad, do you? But this is reality. And the SAFI trial is going on. Nevertheless, this is the market, and this is the world we are working in. This is for lung cancer. You have different options. So I don't know how to lead a multidisciplinary tumor conference in the future. I need someone who's into genetics. I need someone who's in immunology. So it's getting more and more complex. And all in all, we need predictive factors. But do we have these predictive factors? And this is just a calculation example. So we need tissue. Okay, we need blood samples. We need tissues. We need biological informations of the tumor. And these are results of the Breast Cancer Association consortium of 5,000 cases. And every color is one chromosome. And we're looking for SNPs. Okay, we're looking for variations, single nuclear polymorphisms, because we know that these polymorphisms can influence the mutations in the end and can be predictive for mutations. So what do you see here? Nothing. Can you identify the chromosome mutation? No. You need more cases. You need 10,000 cases. And then, do you see more? Yes, maybe. Maybe there. Up there, okay. This is PIK3CA, so this is well known, it's not of interest. You need more cases, 45,000 cases, to get an idea, is there something where another mutation can be detected? And then you get these results. Okay, you have 100 data points for breast cancer risk. You have 101 for early menopause, 90 for osteoporosis, and then the rest is for side effects. And this is the world. So we need to have tissue, and then we need to have big data and data mining, and this is becoming more and more complex. This was done from our German breast group with German studies, and we could identify a new real discovery, but it was done by these tests, so I cannot translate it in English, many tests. So which predictive factors exist for, let's say, bevacizumab, for example? We know this is a VEGF inhibitor, and the FDA in the States forced Roche to do another trial to show an overall survival benefit. The MARIANNE trial was designed, and it was nothing different to the E11200 trial that was led to an approval of bevacizumab. But more interesting, they looked for a predictive factor, and I think bevacizumab is really intensively examined regarding predictive factors. So the patient population having being VEGF-A high, so VEGF-A positive, if you say so, had well, the same benefit, and I go back, as the whole patient population. So there wasn't a predictive factor identified. Sorry. When we talk about immune checkpoint inhibitors, you could say, yes, we measure PD-L1 or PD-1. But this is really new data from ASCO this year, and you see it doesn't matter for triple-negative breast cancer in a metastatic setting. So we treat them regardless of a PD-L1 status. Of course, we measure it, but it doesn't say something to us. What else do we have? When we talk about overcoming endocrine resistance, this is another talk being held in this session. Then we know there is a tyrosine kinase receptor, yes, and we know we can, by a PI3K inhibitor, we can overcome endocrine resistance. We know that we have can target AKT and mTOR complex 1 and 2, and cyclin D and MEK. But do we have predictive factors? No, and no. We have no idea how to treat our patients when we talk about precise medicine. What exists? PIK3A exists. Yes, we have some PI3K inhibitors. First generation, second and third generation. The first generation, we have pan-PI3K class inhibitors like buparlisib. And it's a matter of what do you use when you think of toxicity. So there's a therapeutic index, and you have to have a balance between safety and efficacy. And if you look closer to the BEL-2 study, where buparlisib was combined with fulvestrant against fulvestrant alone, then yes, okay, you had a progression-free survival benefit of 1.9 months with a hazard rate of 0.78. In the PI3K activated group, it was not statistically significant, so the PIK3A mutation didn't add something. But from the circulating tumor DNA, so not from the tumor sample, from the circulating tumor DNA, the PIK3A mutation mattered, and this is something we will take under consideration for the future, that we will use liquid biopsy, not circulating tumor cells. We will take circulating tumor DNA to detect mutations and to treat our patients and so on. So this is a big field and very promising, liquid biopsy. But coming back to the side effects, so buparlisib led to depression in 20% of the patients, and I was participating in the trial, and every patient had to fill out a depression scale, so a depression questionnaire. So these patients should be very stable. In the end, some of these patients tried to commit suicide. So in the end, this is not really balanced when we talk about toxicity and efficacy. So this leads to not a pan class, it led to a single class PI3K inhibitors like tezelizib for the alpha isoform, and there are studies going on with CDK4/6 inhibitor and with a PIK3A inhibitor and letrozole in a hormone receptor positive patient population, and you may think of toxicity in these trials, so I'm not sure whether we will see an approval of these drugs in these combinations, because toxicity is quite high, I can tell you. What about ESR1 mutation? We know from the PALOMA-3 study that it matters, so patients receiving an IBFOR received in a third an ESR1 mutation, not the patients who were treated with tamoxifen before. And regardless of this or independent of this mutation, palbociclib, and I don't want to talk about more because the next talk will come, but this is the mutation issue. Palbociclib could overcome this resistance that was shown with an ESR1 mutation. So we learned our lesson, and we wouldn't give after an AI fulvestrant alone, for example, because we know that we would have an ESR1 mutation. Yes. This is to skip, and there's a signature. And then just to give you, for the last minute, an idea what we do when we don't have mutations. Then we treat our patients in a window of opportunity manner, and then we perform a rebiopsy, and then we measure, in this case, the proliferation index, Ki67. So does Ki67 react? Does the percentage of aggressive cells will go down with this kind of treatment? And this is to be done in different studies. So I will end with this slide to give you maybe a perspective. So this is the Kaplan-Meier curve for conventional therapy. This is targeted therapy I talked about. This is immunotherapy with long-time survivors with endpoints of 1 year of survival, 2 years of survival, 3 years of survival. But what do you want to go for? We want to combine this target therapy and immunotherapy to get more benefit for our patients. Thank you.